As a part of our discussion of microbial genetics and genetic engineering, I'm going to show you how to use a codon chart. For many of you, this is the first time you've even seen one, so it helps to be walked through how we use it. Here is a sample type question of what you would be asked on a quiz or an exam, so let's walk through how to answer it. First off, you read through the question. Say, using the DNA sequence, all of this, what amino acid sequence is formed? And I will always provide a codon chart. So the first thing I would do is I would split this into groups of threes, because if you look over here, codons are in groups of threes, and it makes it a whole lot easier to read. So I've gone ahead and I've gone to put an asterisk in between each of the groups of three. And I double check to make sure that I've written this down the right way, and I have. So the next thing is I'm going to take this DNA and I'm going to transcribe it into an mRNA. So what I'm going to do is for each one of these I'm going to put down what the mRNA would be. So let's go ahead and talk about how we do this. So T always goes with A. That's just how they pair up within the cell. Now when we're going from DNA to DNA, A would go with T, but RNA doesn't have a T. So in RNA, A goes to U, and C always goes to G, and so on and so forth. T goes with A, C goes with G, A goes with U because we're talking about DNA to RNA, G to C, T to A, or C to G, and so on and so forth. So now I've got my mRNA. Now I need to translate the mRNA into an amino acid sequence, or if it's long enough, a protein. So how do I do that? So first, on this codon chart, I'm going to look for AUG. And oh, look, there it is, AUG. It codes for MET, which is an abbreviation for an amino acid, and also all proteins start with MET. So it's also the start of the protein. So I'm going to go ahead and put MET there, and I'm going to get rid of this arrow because it's in the way. Now I'm going to look for AGU. So AGU is over here, and it codes for the amino acid serine, or SER in this abbreviation. So I'm going to go ahead and put that there, get rid of the arrow. Now I'm looking for CAG, it's GLN, GUA is VAL, and this last one actually does not code for an amino acid. It codes for a stop. So there's no amino acid that's going to go here. This codon tells the ribosome to let go of the mRNA and release it so that maybe it can start over at the beginning, but in any case, my amino acid sequence is done. Let's do another sample question. So here I have the same question, I've got the same codon chart, and a different DNA sequence. So I'm going to do the same thing. I'm going to split this sequence into threes. I'm going to double check to make sure that I wrote things down in the right order. I did. And now I'm going to take my DNA and I'm going to transcribe it into mRNA. So same thing here, T goes with A goes with U, because we're talking about mRNA, C goes with G, so on and so forth. Now for this run through, I'm going to pause and let you see if you can find the codon on the codon chart, and then I'll show you where it is. So go ahead and look for AUG. You probably already knew where it was from the previous one. It's right here. Also codes for MET or start. All amino acids sequences, all proteins are going to start with MET. Now look for GGG. Now you may or may not have noticed, but the first letter in each codon is listed over here. So since this starts with G, I'm going to find G, and I'm going to scan through. Oh, there's GGG. So that's codes for GLY. Now look for AAA. and I start over here. Now I can also go over here. Oh, there it is. So it's LYS. And then I'm going to look for AUC. So go ahead and find that on the chart. So I come over here, I'm going to start looking for U's. Oh, there it is right there, AUCILE. And then I'm going to look for UAA. So I come up here to the U's, and oh, there's UAA. That's a stop codon. So there's no amino acid. It just signals the end of my amino acid sequence. While I'm showing you how to use a codon chart, I'm also going to show you how to use the codon chart to figure out another type of question that I'm likely to ask on a quiz or an exam. And that's if there were a change in the DNA sequence, also known as a mutation. So here's an example. Using the DNA sequence, and here's the sequence from the previous question, what type of mutation would occur if I replace nucleotide 6 with a T? So you're probably going, nucleotide 6, what does that mean? Well, it just means you go ahead and count. So 1, 2, 3, 4, 5, 6. So this C is going to be replaced with a T. So what kind of change is that going to make? So the first thing you need to do is figure out what would happen if there wasn't the mutation. So I've already walked through how to get that. So now we're going to talk about what the change is. So this T, I'm going to bring it down here to my spaced out DNA. And so what change is going to happen here? Because G doesn't go with T. What does go with T? Well, an A does. So now I'm going to look for GGA and see if there's a change from the glycine. So let's look over here. Let's start over here. I'm looking for the GGA. Okay, it still codes for a GLY. This is known as a silent mutation. In other words, there's been a change in the DNA sequence, but there's been no change in the protein. So when a mutation has occurred, it just doesn't show up in the protein. So let's take a look at a different kind of mutation. So same DNA sequence, but this time we're going to change nucleotide 8 to a G. So let's go ahead and count. 1, 2, 3, 4, 5, 6, 7, 8. We're going to change that to a G. Now I'm kind of covering up 2 there, so we're going to go ahead and move that down here. 2, 3, 4, 5, 6, 7. Okay. So now, once again, I'm taking what the original sequence was, and I'm going to see what change occurs. So G doesn't go with A, but C does. So I'm going to look for ACA and see if it's still LYS. So I'm going to come over here to the A's. I'm looking for ACA. There it is. Oh, there has been a change. LYS has changed to THR. So this is what's known as a missense mutation. There's been a change in the DNA that results in a change in the amino acid sequence, but only one amino acid. It's kind of like misspelling a word. So think of it that way. Missense, misspelling. Let's take a look at another one. So here we've changed nucleotide 7 to an A. So we're going to go 1, 2, well, let's go down here where they're spaced out better. 1, 2, 3, 4, 5, 6, 7. Okay, so this first T and this T are going to change to an A. So A doesn't go with A. A goes with what in mRNA? A U. So let's look for UAA. Let's see, we're looking along. UAA. Oh, we have an early stop. So it doesn't code for an amino acid, it codes for stopping before it should over here. We call that a nonsense mutation. That's like if I were to just suddenly stop speaking, my sentence would not make sense. Now, depending upon where this nonsense mutation occurs, it can occur at the beginning of a gene and kill the gene. It no longer makes a functional protein. It can happen later on where it has less of an effect, but still may have an effect. But in any case, it is a nonsense mutation. Now let's go on to our last type of mutation and how you would figure that out on a quiz or a test question. So let's say that nucleotide number 2 was deleted. So this one is going to get deleted. So what I'm going to do is I'm going to rewrite this with this missing so that I can compare it up here. So this A is gone, so I've just shoved everything on over. So this C comes over here, this C comes over here. I'm separating it now. So this is the new sequence. We call this a deletion mutation, but let's see what kind of deletion mutation it is. So I'm going to take my mutated mRNA and I'm going to transcribe it into a mRNA, and I have a dash here because I don't know what else is on the chromosome. So let's go ahead and see what kind of changes we have. So the first thing is we're going to have an ARG. We're no longer starting with MET. If this were to happen, the gene would not start. It would probably not be transcribed because there's no AUG or TAC to tell RNA polymerase that, hey, here's the start of a gene. And then the ribosome would also not know where to start because there's no AUG. And so we call this a frame shift mutation. Everything has been shifted one way or the other. And this happens with deletions, or if I were to happen to add one in. Let's say I added a T in here, that would shift everything that direction. So those are the types of mutations that I want you to know, and this is how you use a codon chart. Make sure that you ask me if you have any questions. I'm happy to answer them.